to this conference, and I like it very much. So I'm really happy to be here. I was somehow appointed a bit less time than other speakers, but I have the permission from the organizers to use some extra 5 minutes. So if I'm late, it's not that I'm stupid, but anyway, I had the permission to get a bit more time. So I'll show you some of our recent results on the quest for the synthesis of hypermodified nucleic acids for targeting proteins. So having so many speakers on similar topic already, so I can skip most of the introduction, but just very briefly. The traditional medicinal chemistry has almost exploited its potential because, like all the enzymes or proteins that have binding sites for small molecules are already explored, and the biologicals typically are used only on extracellular targets. While most of the proteome is not druggable by small molecules, or at least not easily druggable by small molecules, because they are typically involved in protein-protein or protein-nucleic acid interaction. And for this audience, I don't have to stress that in principle these interactions could be targeted by nucleic acids, but like classical nucleic acid aptamers are difficult to target hydrophobic proteins, and we've seen excellent talks by Larry and Nebolisha and also by Michal Hocek that by chemically modified aptamers you can do more. But in my lab we are trying to go even further, and we are trying to develop an approach to synthesize hyperfunctionalized nucleic acids, where basically every single base would be bearing some modification, some small molecule attached to it, and to be able to target basically any protein like, and basically use the DNA just as a scaffold for display of small molecules for different purposes. And the chemistry is extremely straightforward, because we can use like late stage functionalization of nucleoside triphosphates. That if we have a halogenated nucleoside triphosphate, we can perform all sorts of cross-coupling reactions, and in one step we can attach some aryl groups and alkynyl groups and some other modifications, and we can even reduce the alkyne to alkyl, so to have flexible linkers. So basically, and we can use the click chemistry as it was shown by Marcel this morning. So having this chemistry in hand, we can attach the modification in one step, and then in the second step use the polymerase. And again, I don't have enough time to go into details, but we can use all sort of methods using DNA polymerases, which allow us to incorporate one modification or several modifications, or even like large density of modifications using PCR. And using ribonucleoside triphosphates, we can use either T7 RNA polymerase to synthesize modified RNA, or we can also use some engineered DNA polymerases that can also synthesize RNA. But I'm not going to talk about this topic today. So we can easily use all the modified nucleotides in the synthesis of modified DNA or RNA. And now I will focus on specific modifications that can be used for targeting proteins, and I will show some examples that will basically detect protein binding, then some examples of modifications that can cross-link to the interacting proteins, and then we will slowly go into the hypermodified nucleic acids. So just to detect protein binding, we can use all sort of fluorescent modifications that are environment sensitive, and basically we use two principles. We have many examples from each principle. I will show just one example of each. We can use some molecular rotors that are basically viscosity sensors, and if we modify DNA with such body molecular rotor, itself, it has bright fluorescence, but very short lifetime. And when a protein interacts with these DNA probes, then the lifetime is significantly extended, and we can use it even in the slim microscopy to detect protein and nucleic acid interactions in live cells. And the second principle, which we often use, is the solvatochromism. We have number of solvatochromic fluorophores. This is just one example, but we have all sorts of colors. And here, for instance, with DNA modified with this modification, is almost non-fluorescent, but it is like very weak red fluorescence. But if the DNA interacts with protein, then the polarity of the environment around this modification is changed, and we can see significant enhancement of fluorescence and color change. So and this can be also used in live cell, and we can even use these building blocks to get incorporated in the live cells. But to be able to do so, we need a transporter. And a colleague from our institute, Tomáš Kraus, developed a transporter based on cyclodextrin attached cationic peptide that binds nucleoside triphosphates and transports them across the cell membrane. And then in the cell, there is high concentration of ATP, which releases our modified nucleotide. And if it is a substrate for polymerases, then it gets incorporated. And indeed, we can use this. So, for instance, with this brightly fluorescent nucleotide, these are some cancer cells treated just with the nucleotide, and you can see that there is nothing much inside. There is some staining of the membrane. But if we use the transporter within 3 minutes, all the fluorescence is inside, and if we wait a bit longer until the cells get into the S phase, then they start to synthesize DNA, and we can see nice staining of the genomic DNA. And we are trying to use this approach for imaging of DNA and RNA synthesis and DNA and RNA interactions in live cells. Just yesterday, our paper on this was published. I did not want to talk about it because it was a poster at this conference, but there was no proper poster session, so I will just mention it. And you can look into the electronic materials that you should have for details, and it was just yesterday published online in Bioconjugate Chemistry, where Ambras Pampinato, who is in the audience, was able to synthesize the whole bunch of nucleoside triphosphate modified with different TCP or VCN reactive groups with different linkers. And she tested different tetrazenes, and she found some suitable combinations that really work for metabolic labeling of DNA synthesis in live cells. So for details, you can look at this paper. Then the second thing what we can do with modifications is to cross-link to the interacting proteins, and then in principle, if we have some aptamers, we could make them like permanent covalent inhibitors of the target protein. So we need some functional groups that would be stable enough for the DNA synthesis, and then reactive enough to react with certain amino acid side chains for making the cross-link. So the first one was this vinyl sulfonamide that survived the polymerase synthesis and cross-linked to DNA interacting proteins that have cysteine in the proximity. Then we use this chloroacetamide that worked in a similar way, and it's cross-linked with either cysteine or histidine. And again, only when the proximity effect was there. So, like if there was a protein having many cysteines that did not interact with the DNA sequence, the cross-link was not there. But like cysteine and histidine are not so common in DNA binding proteins. So certainly the next targets were lysine and arginine. For lysine, we used the squaramate linked nucleoside triphosphate, and it works just beautifully, and without any additional reagent, it cross-links to lysine-containing peptides and proteins. And for arginine, we can use either glyoxal or 1,3-diketones that also can nicely form the cross-link through formation of pyrimidine ring. And then we can apply the same principle for RNA. So certainly the synthesis is a bit more difficult, but for instance, this chloroacetylamide linked ribonucleoside triphosphate is used for in vitro transcription with T7 RNA polymerase to synthesize modified RNA, and then we were able to cross-link to some RNA interacting proteins like who are HIV reverse transcriptase or human argonautic protein, and like by partial digestion of the protein, we were able to identify what was the binding mode to the RNA. So now let's move from one modification to more modifications. And here certainly the goal is to have fully modified DNA sequence where every nucleotide would bear a modification. And the goal here would be to have basically aptamers or small oligonucleotides that would target basically any protein or in principle any biomolecular interaction. And here certainly the challenge is to be able to enzymatically synthesize this, because it is already heavily modified, and also then to be able to sequence it, because it's not obvious that you can easily sequence such a heavily modified oligonucleotide. So to test this, we started with this set of modified nucleoside triphosphate. So basically, you can see that we use all 4 nucleobases, and we attach 4 hydrophobic modifications that somehow resemble some amino acid side chains, and we attach them either through rigid alkyne linker or through flexible alkyl linker. And then we tested whether we can synthesize the fully modified strand. And now the answer is yes, we can do it. We can do it. The alkyne linked nucleotides are better substrate than the flexible alkyl linked nucleotide. With the alkyl linked nucleotides, we can have something like 35 to 40 consecutive modifications, and then basically the DNA becomes totally insoluble in water, and that's the end. But with the alkyne linked nucleotides, we can go quite long. So we went as long as 150 consecutive modifications, and it still works. And we can combine the alkyl and alkynyl linked nucleotides as well. And then we also studied the hybridization. It still hybridizes and secondary structures. It is all quite interesting, but I don't have time to talk about it. But I will address one thing which is like commonly underestimated in similar studies. You know, there are many papers in the literature around where the authors claim that they run PCR with heavily modified nucleoside triphosphate, and what they show is that they have a band on agarose gel, and then they like isolate the gel, the band, and they re-PCR it and sequence it, and then they say that it all works. It may work, but this is not the proof that it works. Because like if you want to prove that you have PCR, then it must be like the exponential amplification, and you need to show that both reverse and forward primers are extended. And to test it, the only way how to really prove it is that you label each of the primers with a different dye, and then if you have the spot on the gel, then it should be visible with both wavelengths. And So we tried different combinations of 2 modifications, 3 modifications, and 4 modifications in PCR. And as you can see, we had the FAM channel and sci-fi channel, and to show successful PCR, it should be visible in both. And the combinations of 2, they nicely show that you can see them in both channels, whereas the combinations of 3 or 4 modifications, we see only the blue channel, which means that it is only asymmetric PCR. It is just the linear amplification of the forward primer, not of the reverse primer, which means, in other words, that the heavily modified strand is a poor template for synthesis of another heavily modified strand, which makes also difficult to sequence it. And then now the question is whether so we can synthesize one modified strand, long one, and then the question is whether we can sequence it. But fortunately, we can re-PCR it with non-modified nucleotides, which is easy. But again, we have to be sure that we amplify the modified strand and not the traces of the original non-modified template that was in the mixture. So we had to develop this trick that the original template contains on the 3' end this group, which prevents its extension in this direction. And then we use the primer that has 5' overhang, and we run the asymmetric PCR to synthesize this modified strand. And then for re-PCR, we design the primer which is complementary to this part, not to the original one. And only using this, we can ensure that we did not amplify the original non-modified template. So, like, nobody else is doing this, but I tell you, this is quite important to be sure that your heavily modified DNA is what you claim it is. Okay. Then we want to have some better methods how to synthesize single strand fully modified oligonucleotide. So we are thinking about using RNA template because you can easily digest RNA. So we tried to employ RNA template and DNA primer. We tested several reverse transcriptases, and none of them worked with the modified nucleoside phosphate. But we tested DNA polymerases, and they work beautifully. For instance, BSD330 worked perfectly, and it can synthesize fully modified DNA from RNA template. And now, so we have now the single strand after digestion with RNAs. We have single-stranded DNA full of modifications, but still the primer is there and the primer is not modified. So can we somehow easily get rid of the primer? So we were thinking, why not using even RNA primer? RNA template and synthesize DNA nodes. So I can tell you this doesn't work. But what we can do, frozen. We'll start with.
I check out the presentation mode. Soon to be back in a minute. We've had that a couple of times.
This don't. Overhead next time.
Yes, overhead projectors and
Yes.
So or should I stop sharing?
Let's try that. Yes,
Yeah, but it's the problem with this. It takes several minutes. It's very encouraging. Okay.
We can
I could ask to help, but I'm not sure if I can fix it.
By the time this is fixed, they probably publish 2 more papers. To the 5 minutes.
Share the screen on the shoulder. It's not easy for there at all. Excellent. Anyway, so as I mentioned, the extension of RNA doesn't work, but we can use DNA primer with the ribonucleotide with E prime end, and we can use DNA primer with the ribonucleotide at the end and somewhere in the middle, and this works. So we can synthesize these types of hybrid oligonucleotides that contain 1 or 2 ribonucleotides in the sequence, and then we can use RNAs that will cleave the RNA template, and it will cleave the 2 ribonucleotides to make very short oligonucleotides, and we can generate fully modified polymer that contains only the modification. The problem is that we don't see it because we have lost the fluorescent label. So we can use TDT and another fluorescent nucleotides to add it at the 3' end to visualize it. But we had it even before. So I think this is quite a promising approach that we are now trying to use to generate libraries of sequences and try to do selections. So far we do not have any fully modified aptamer. We have just accepted paper on our first modified aptamer with only 1 modification. We have used this flexible hydrophobic modified DHEP that was used in the selection of aptamer against heat shock protein 70, and it's kind of worked. You know, after seeing the presentation from the 2 presentations from Somalogic, I should even not show it, but we have 1 other. So that's probably it. But we can like use other combinations of modifications, and for instance, we have combinations of 4 different sugars like mannose, galactose, glucose, and galna, and we can generate sequences like decorated by different sugars, and then we are trying to use them to target some lectins, and with concanavalin A we had like sequences modified with just 1 sugar and sequence modified with all 4 sugars, and it bound better. But this is not like specific aptamer with certain sequences. This is just with combinations of sugar, and it works better than just some sugars. We can actually use the fully modified DNA also for some other purposes. And one other purpose that we are pursuing is the redox coding of DNA bases. And here we again generated 4 different nucleotides, and now each of them is bearing another like different redox label with a different redox potential. So we had carborane, then metallocarborane, ferrocene, and the fourth modified base was 70-AG, which is also electroactive. And then we run the primary extension on this template that contains a spacer and a tail sequence that is used for attachment to electrode. So we have electrode that has capture probe attached, and then after the primary extension, we can hybridize with on the electrode, and then we can measure square wave voltammetry. And now we have 4 different labels that are orthogonal, and simply by measurement of 1 voltammetry, we can say from the ratio of the 4 signals what was the ratio of the A, T, B, and C in say the unknown sequence of DNA, and we are trying to use this further, hopefully for some simple sequencing on electrode with just electrochemical readout. We can also attach some lipids. Several talks actually have been using oligonucleotides with cholesterol or some other lipids as one of the ends of the oligonucleotide. We have synthesized a number of lipid-linked glucoside triphosphates, and we tested them as substrates for DNA polymerases. This was already quite challenging, so I can tell you that the phospholipid ones did not work at all because kind of they interact with the polymerase too much, and never able to get the modified DNA. But with cholesterol and palmitate, we were able to synthesize different sequences of DNA having 1, 2, 3, or 4 modifications, and we could even combine them. And we have compared them for like anchoring of the oligonucleotides into membranes, and we found that if you have 2 cholesterols or a combination, this is not the last slide, 2 cholesterols or a combination of cholesterol and palmitate separated by several nucleotides, then the anchoring to membranes is very efficient and also persistent. So that was the last scientific slide of my talk. So I hope that I've shown you that the modified glucoside triphosphates have a lot of potential for synthesis of different modified nucleic acids, and we can even make hypermodified nucleic acids and hopefully make some of them useful. And the last thing what I need to do is to thank all my coworkers and you all for your kind attention.